Good day everyone! So today I will be discussing to you this branch of biology which is the formology, physiology, bacterial taxonomy, and bacterial genetics. So let's start. Organisms need cells to live and survive to the changes in the environment. Even the tiniest microbes and bacteria has its own cells to regulate and control their features for survival. Have you ever thought what a disaster it would be if we failed to study or recognize microorganisms that plays a crucial role in our life? Bacteria are considered to be simple compared to other organisms, as there is no nucleus or membrane-bound organelles. It is a single cold microbes. Bacteria have an extra circle of genetic material called plasmid that contribute to the advantage of the bacteria, and it contained a single loop DNA. So speaking of DNA, this include the prokaryotic cells and eukaryotic cells. Prokaryotic cells include bacteria and archaea. Their DNA is not contained within a nucleus. Instead, their DNA is circular and can be found in a region called nucleoid, which floats in the cytoplasm. So, eukaryotic cells are found in plants, animals, fungi, and protists. Their DNA is contained within a membrane-bound nucleus. Eukaryotes are organisms containing eukaryotic cells. Bacteria are classified into 5 groups according to their basic shapes: the spherical, the rod, the spiral, or the corkscrew. They can exist as a single cells in pairs, chains, or clusters. So bacteria are classified into 5 groups according to their basic shapes. These are the spherical, the rod, the spiral, the and the corkscrew. So they can exist as single cells in pairs, chains, or clusters. So to help us understand more about bacteria, let's tackle about the bacterial morphology and physiology. So first is the bacterial morphology. So what is morphology? Morphology came from the Greek word morph, meaning shape, and logos, which means study. So it is a branch of biology that studies the form, the structure of organisms, and their specific structured features. This encompasses the external, such as the color of an organism, the shape, the size, and pattern, and also the internal structure, which are the features, internal structures, like the bones and organs of an organism. So, second is the physiology. Physiology is a branch of biology that studies the normal functions of organisms and their parts. So the variation of bacterial cell shapes is often underappreciated. So bacterial morphology and physiology is extremely diverse. Specific shapes are the consequence of adaptive pressures. Optimizing bacterial fitness, shape affects critical biological functions including nutrient acquisition, motility, dispersion, stress resistance, and interaction with other organisms. Although the characteristic shape of a bacterial species remains unchanged for vast numbers of generations, periodical variations occur throughout the cell division and life cycle, and this variation can be influenced by environmental conditions. So next is the bacterial taxonomy. Bacterial taxonomy is a branch of the field of morphology and encompasses its 3 key elements: the characterization of identification of names, the classifications of the type of organisms, and the nomenclature or naming of microbial isolates, which are the basis of a formal proposal to describe new species. It also includes the bacterial classification that is defined as the arrangement of organisms into taxonomic group or the taxa, and the basis of their phenotypic or the observable, and the genotypic or their genetics, similarities, and differences. It allows proper and systematic grouping of microorganisms. So organisms are classified into three main kingdoms: the animals, the plants, and protista. The protista contains unicellular microorganisms including eukaryotes and prokaryotes, although no universally accepted bacterial classification system is available. So three main approaches are usually followed. These include the phylogenetic. So phylogenetic is the classification of the type of hierarchical classification because it denotes an evolutionary arrangement of species. One characteristics being employed for divisions at each branch or level. So, one characteristics being employed for division at each branch or level are related on evolutionary basis where several groups are used such as divisions, the classes, the orders, the families, tribes, genera, and the species. So next is the Adansonian. So Adansonian is a classification that gives equal weight to all measurable features and groups of bacteria and the basis of similarities of several characteristics. Then, Adansonian classify the characteristics of a bacterium, facilitates the identification of several broad subgroups of bacterial strains that are further subdivided into species. So this type of classification is based on the properties of large number of properties is known as numerical taxonomy. And lastly is the genetic classifications or molecular classifications. Is based on homology of the DNA-based sequences of the organisms. DNA relatedness of the microorganisms is tested by studying the nucleotide sequence through hybridization or combination and extracting DNA from the organisms to be studied. Then, next is the bacterial genetics. So, what is bacterial genetic? Bacterial genetic is a branch of molecular biology that provides powerful tools for deciphering the understanding about genetics, genomes, and genomics that serves as the function and pathway organization of cellular processes. So this involves both discovering the nucleic acid and the functions and sites that control regulatory genes and sites that control individual gene expression and determining which genes are co-regulated with its ribonucleic acid or the messenger, the ribosomal or the transfer RNA, and thus likely to participate in the process protein synthesis. Gene expression also takes part on the process of transcription and translation, that often genes that are co-regulated are located next to each other in the same transcriptional unit or the anoperon. But there are numerous cases as dispersed sets of genes that are co-regulated or regulons. So, in conclusion, this evidence that morphology and physiology, bacterial taxonomy, and bacterial genetics ensures development of aberrant upon bacterial mutations and cycles. So, over generations, this branch of biology maintain an active process guided to robust regulatory circuits that dictate interactions between a bacterial cell and its environment. Therefore, morphology, physiology, bacterial taxonomy, and bacterial genetics should be viewed as a major evolutionary and adaptive process that contributes greatly to prokaryotic ubiquity and versatility. So that should be all. Thank you for listening.